Awesome. Well, thank you for the introduction and for the opportunity to speak here. So I'm excited to tell you about my work doing haplotype reconstruction with low-pass WGS data. And today I'm going to primarily talk about the DO. And as we've covered extensively over the last few days, all of these complex mouse and rat populations are great and premier genetic resources for complex trait genetics, and the DO is among them. And I really like this illustration in a recent paper by Michael Saul showing the relative utility for each of the CC and DO founder strains, extant CC mice, and the diversity outbred population. And one of the things we're interested in doing is increasing community uptake and increasing accessibility to this population. And one of the limitations we're really interested in addressing is the fact that DO mice are non-fungible, meaning that they have to be genotyped each time they're phenotyped. And addressing these gaps will accelerate QTL refinement, hopefully, and also bring a cost reduction as well to this resource. Typically, these DO mice are genotyped on the Gigamuga array, which has roughly 140,000 markers. And at this time, I'm just going to point out, if you follow this QR code, I've done a little bit of QC work, moving some of the Gigamuga marker annotations and CC and DO reference data over to the GRC M39 mouse genome, the new build. So follow that QR code if you want, take a look and see if your positions have changed, any founder genotypes have moved, what have you. And I know that there are a couple sample mix-ups in the reference data that were fun. So take a look at those, you can poke through those. So yeah, for DO mice and for any other mouse from a complex cross you want to genotype, it runs you about $100 a sample. And another limitation we've observed is that we believe we're missing crossovers. So this is from generation 36 of the DO. And these data were processed by Howe at the JAX CSNA. On the x-axis, I'm showing the haplotype block size obtained from haplotype reconstructions from these mice. And we can see that smaller than 0.5 to 0.25 megabases, we have a drop off of recombination blocks that we observe in the data. And when we track those observed autosomal crossovers across multiple DO generations, we observe that we have approximately 12 new crossovers per generation. And some quick back of the envelope math indicates that that's about half of what we expect per generation. So what we're really interested in doing is capturing these missing recombination breakpoints. And one way we can go about reducing costs and adding resolution is by finally pursuing a whole genome sequencing approach to genotyping the DO mice. So the goal for this initial pilot study was to provide a competitive cost to investigators that runs them less than $100 a sample with comparable or better accuracy or precision compared to the Gigamuga array, and also implementable as a service way down the line. We have a couple different pilot data sets for this. The set I'm going to talk about primarily today is a set of 48 DO mice from generation 41. And we pursued 3 different genotyping methods. 2 of them are sequencing based. We did a dd-rad-seq approach and a sequel library prep kit, which uses a TN5 transposase to randomly insert throughout the genome, producing a sprinkling of reads effect rather than a stacking of reads. And we also genotyped these mice on the Gigamuga. And all of this was done with the help of Genome Technologies. And these mice came from these samples were actually cell lines from Laura Reinhold. And we also have a set of 48 four-way cross mice that are in the F2, F3 generation. And the only reason I bring this up is because we're hoping to keep this method flexible to other diverse mouse crosses. These strains are wild-derived strains from the Mus musculus musculus and Castanea subspecies. So while these mice don't have the same number of recombinants across the genome, obviously, they do have very different variant structures. So we're hoping to have that extended as well. Here's the pipeline we're using. You don't need to ingest any of this in any depth at all, but I just want to point out a couple quick things. One of which is that the engine of this workflow is genotype imputation fueled by a software called Quilt, which uses founder reference data to impute genotypes given low-pass whole genome sequencing in a reference data set. And with the DO, we're fortunate to have good reference data, as Robin talked about today. Where there are a couple different covariates that are provided, but the main takeaway here is that the input is these demultiplexed FASTQ files from each sample, and the output is cross objects and haplotype reconstructions in the form of allele and genotype probabilities that can be passed directly into RQTL2, with the obvious application downstream of potentially doing some mapping with these data. And the last thing I want to mention that I'll come back to is that we can also supply a downsampling parameter because we'd like to see what the point of dropout is. How low pass can we go, essentially, and still obtain an accurate haplotype reconstruction. So here what I'm showing is the founder haplotype composition across 2 representative chromosomes. And so on the y-axis is the percent of samples with a given founder allele at the location, and on the x-axis is genomic position. And the only takeaway here that I want you to see for now is that when we compare the quilt haplotype reconstructions to Gigamuga, we see that they're basically reflections of each other. And if you a savvy observer will also notice that we recapture the depletion of WSB alleles on chromosome 2 after the purge of the R2D2 locus after generation 12 of the DO. We can look at karyograms from these mice. So what I'm showing here is on the x-axis is a stacking of the allele probabilities for each locus, and on the y-axis is genomic position. So this is a little bit different than the traditional representation of a karyogram, which is typically done by inferring the most likely genotypes at a locus as is the default output from QTL2. Here we're showing the actual vector of allele probabilities. And for the most part, we see that the genetic structure from the sequencing-based approaches and the Gigamuga-based approaches are mostly conserved, with some important variations that I'll talk about later. But so here is the sequel data, which uses the TN5 transposase, and the dd-rad-seq roughly mimics that as well, with some variations as well. So this was really encouraging to see. And at this point, I just want to remind everybody that we're doing genotype imputation, but not all of the. So one, for speed's sake, we really try to filter down these imputed variants so that our QTL2 doesn't crash when we do the haplotype reconstructions. But I do want to point out that the number of variants we get from imputation and their quality is sensitive to the coverage of the data that we put into the pipeline, which is intuitive. But when we go and compare the vector of allele probabilities at each Gigamuga marker to its nearest marker in our sequencing-based pipeline approach, we see that 0.05x coverage is roughly equal in concordance to the full coverage that we obtained if we just retained every single alignment from each sample. So this is really encouraging to see, and we have that recapitulated in dd-rad-seq and the sequel data. There is that little bump of points in the dd-rad-seq data at full coverage. Those are all from one sample, so there might have just been a sample quality thing there. And if we just do a rough calculation based on 0.05x coverage and what we estimate the library prep cost to be, so excluding labor and shipping or whatever is required, that just in terms of genotyping the mouse and prepping the DNA, we see a substantial cost reduction. So we think we're on our way there, but there are still obviously some improvements to make. And in terms of recapitulating or recapturing missing crossovers, what I'm showing here is the Gigamuga crossover count compared to the crossover count we obtained from both sequencing-based approaches. And we see that coverage is a main driver of this, but with 0.05x coverage, we obtain roughly a 2-to-1 relationship between the quilt crossover counts and the Gigamuga crossover counts. And that's important because we estimate that there were roughly 300 crossovers in generation 7, and adding 24 new crossovers for generation, which is what we estimate we should expect based on the basic Mendelian genetics. We expect roughly 1,100 crossovers in the generation that we're analyzing, and we see that the majority of samples are clustered right around that point. So this is a really encouraging sign. And one quick thing I do want to point out is that for the concordance plot that I showed before, it was the median concordance across the chromosome, but there is a lot of local variation across the chromosome. And when we look at tracks where we can look at allele sharing across the different founder strains of the DO, we see that a major driver of discordance is these known spots where the classical inbred strain founders that contribute to the DO are actually identical by descent. So when we look at this region, we can go to one of the karyograms where these genotypes are discordant, and we see that where the Gigamuga uses a little bit different of an approach and really sells out on the B6 het region in that area, we have a lot more conservative spreading of those probabilities for the alternate allele in the allele probabilities. So this is something we're looking at and seeing if we can determine how much of it is false positives versus real stuff that we need to remedy in our estimations. So for future work, we want to continue to evaluate support, like I said, make sure we know that we're actually picking up the false or picking up real recombination events, and also determine whether newly identified and characterized structural variations in the CC founders can explain this discordance. We would also really love to test empirical data. So get a bunch of DO mice and do some, pretty high powered, but just enough power to do some QTL mapping and see if our peaks actually move with these new haplotype reconstructions. And we also would like to evaluate the possibility of long read sequencing as it becomes cheaper. With that, I'd just like to acknowledge everybody that contributed to the projects, all of their funding, and specifically Dan Gaddy and Lydia, who were who provided a lot of guidance, and Lydia did like all of the prep and all the sequencing with the help of the Genome Technologies course. So with that, I'll take all of your questions.